Alright, so in this video we are going to talk about two very important pathways, two very important signaling pathways. One is the Wnt beta-catenin and the next one is the hedgehog pathway. Now these two pathways are very important in some of the developmental process. So developmental process and organization of structures in a lot of organisms, for example in Drosophila melanogaster. Now without these two signaling pathways, a lot of the processes, a lot of the developmental processes are halted, and the organism is not formed properly. So this Wnt pathway and the hedgehog pathway are very important in those aspects. We'll start off with the Wnt pathway. Well, for Wnt beta-catenin pathway, the ligand is Wnt. So this is a protein which is the ligand for the pathway. So Wnt, let's say this is Wnt, and this is received by the cell that is having the receptor for Wnt. So let's say this is my receptor for Wnt. This is received by that receptor, and it relays some signal inside the cell, which sort of helps to activate a protein called beta-catenin, in short beta-cat. Now if Wnt is absent, if Wnt is not there, then beta-catenin activation is halted. We're going to talk about that, how the beta-catenin is activated and what happens when Wnt is not there. So the ligand is Wnt, and the functional pathway through which it works is by activating another protein which is called the beta-catenin, or beta-cat in short. Okay, so we have two sets of pathways over here. We're going to divide that and we're going to talk about it separately. Now in the left we have the situation where Wnt is absent, so Wnt is not there. What happens then? So this receptor, which is called frizzled, this is the receptor for Wnt, and this receptor sort of takes in Wnt and relays the signal. But when Wnt is not there, the signal is not relayed, and we have another protein which is called disabled, this protein, which is in its inactive state when Wnt is absent. So in absence of Wnt, no signal is relayed, and this protein disabled is in its inactive state. Now what happens, since this is in its inactive state and we have the beta-catenin over here, so beta-catenin is present inside the cell, and this beta-catenin, as you can see, it is surrounded, or it is ligated to a lot of proteins, right? So it has got this CK1, which is a kinase, has got this GSK3, which is again a kinase. So CK1 stands for casein kinase, and GSK3, this stands for glycogen synthase kinase. So GSK3 and CK1, these two are kinases, and these are held up by two other proteins which are AGSine and APC. So when all of these proteins are holding beta-catenin, then these two kinases, CK1 and GSK3, they can phosphorylate beta-catenin. They can put phosphate groups on the beta-catenin, because when this whole complex is being formed, the casein kinase, that is CK1, and GSK3, these kinases are active, and they can phosphorylate this beta-catenin. And since they phosphorylate this beta-catenin, the beta-catenin becomes unstable. So phosphorylation of the beta-catenin makes it unstable. Now what happens, as the beta-catenin is unstable, it is sent into a complex which is called the proteasomal complex. And proteasomal complex degrades this beta-catenin molecule. So since beta-catenin gets degraded, the downstream signaling is halted. Now, as this beta-catenin is unstable, what happens? This beta-catenin this beta-catenin goes through a process which is called ubiquitinylation. And when something is ubiquitinated, this is destined for proteasomal destruction. So again, from the start, this whole complex, which is comprising of CK1, GSK3, axin, and APC, this whole complex is holding beta-catenin, and since this complex is formed, these two kinases, the CK1 and the GSK3, these two kinases are active and they phosphorylate the beta-catenin, which in turn makes the beta-catenin unstable, and that leads to ubiquitinylation of beta-catenin, and that ubiquitinylation, it sends the beta-catenin for proteasomal destruction. Now, since beta-catenin is destroyed, it is no more present, it cannot move into the nucleus and activate it cannot activate the transcription factors that are related to the developmental processes. Now let's see what happens when Wnt is present. When Wnt is present, this Wnt is received by the frizzled receptor, which in turn gets activated. So receiving the Wnt makes the frizzled receptor activated, and this binding is made more strong by the use of another co-receptor, which is LRP. When LRP binds to this frizzled receptor, it makes the binding of beta-catenin to frizzled very strong. Now, in this case, disheveled, which was present inside the cell, it was in inactive form. Now, when Wnt binds, this frizzled receptor can recruit this disheveled protein. So this disheveled protein comes and interact with the frizzled receptor. And as the disheveled protein interacts with the frizzled receptor, it becomes active. Now, activation of disheveled recruits all the proteins that were holding beta-catenin. So, for example, disheveled recruits axin, and axin recruits GSK3 and CK1. So, CK1 and GSK3, these two comes to to the binding point of axin and disheveled, and phosphorylates the LRP and frizzled complex. So disheveled is recruiting axin, axin is calling on to GSK3 and CK1, and now as these proteins are getting called upon by disheveled, APC is no more APC is no more binding onto the complex, so APC is made free, and APC is made free, so beta-catenin is also made free, and now beta-catenin is stable, because all of the proteins that were holding beta-catenin are now called upon by the active disabled complex, and they are coming and phosphorylating this LRP frizzle complex. And since this LRP frizzle complex is phosphorylated, this makes the binding much more stronger. Now beta-catenin is free, and it can relay the signal that it is supposed to do. It moves into the nucleus, and it binds to a transcription factor which is called LEF1 TCF factor. So this green color, this is the LEF1 and TCF factor. So as it binds to this LEF1 TCF factor, there was already a protein called GROCHO, which was inhibiting the activity of LEF1 TCF factor, as you can see over here. Now, as beta-catenin binds to LEF1 TCF factor, this GROCHO, there is a conformational change in LEF1 TCF factor, and this GROCHO is, it is made free. So the LEF1 TCF factor, it changes its conformation and lets go of the GROCHO that was inhibiting it. Now, as I said, this LEF1 TCF factor, this is a transcription regulating factor. Now it can move on to do the transcription of the required genes that are supposed to be transcribed because of the Wnt signaling pathway. So that was all about the Wnt signaling pathway and what happens when Wnt is absent or Wnt is present. Now moving on to the hedgehog signaling pathway. For this, the ligand is hedgehog, and it works by the activation of a protein which is called smoothend. So Wnt was activating the beta-catenin, and for hedgehog signaling pathway, it is activating the protein called smoothend. Now let's see how it happens. So starting off with the hedgehog signaling, we are going to see the hedgehog signaling in a similar way like we saw for Wnt. We're going to see what happens without the hedgehog signal. We are also going to see what happens when the hedgehog signal is present. Now, when the hedgehog signal is absent, the hedgehog receptor, which is on the cell, that is the patched receptor, this one, this receptor inhibits a protein called smoothend, which is present on a vesicle inside the cell. Okay, so in the absence of the hedgehog signal, the receptor, which is patched, it keeps the protein smoothend in its inactive state, which is present in a vesicle inside the cell. So since this is inactive, smoothend cannot perform the downstream signaling that it was supposed to do. So what happens, there's this protein called cubitus interruptus, or CI. So cubitus interruptus. This cubitus interruptus, CI, is held on to by two proteins. The first one is fused, and the next one is costal 2. Now this costal 2 and fused, when it is holding on to this cubitus interruptus, it is recruiting a lot of kinases. For example, the pKa, the GSK3, and CK1. Right? So fused and costal 2, these are recruiting the kinases, which in turn phosphorylates this cubitus interruptus. You can see over here the phosphate groups present on the cubitus interruptus. Now, as these phosphate groups are put on to the cubitus interruptus, like beta-catenin in the Wnt pathway, this is destined for proteasomal destruction. So it is sent to the proteasomal complex, and what happens is that after the destruction, there are two subunits or two destroyed parts of the cubitus interruptus. One is the small subunit and the other one is the large subunit. Now, when this happens, when the cubitus interruptus is divided into these two subunits, the small subunit moves into the nucleus, where it acts as a repressor for the genes that were supposed to be activated by the hedgehog signaling. So once again, the cubitus interruptus, once it is phosphorylated by PK1, GSK3 and CK1, it is destined for proteasomal destruction through the process of ubiquitinylation. Now, once it goes to the proteasomal complex, it is destroyed and made into two different sized subunits. The small subunit, which is formed after the destruction, can move into the nucleus and can act as a repressor, which represses or inhibits the genes from transcribing, which was supposed to be transcribed by the hedgehog signaling. So that is what is happening when the hedgehog signaling is absent. Now, when the hedgehog signal is present, what happens? This hedgehog molecule over here in red, this hedgehog molecule is received by the receptor that is patched, and this binding is made more stronger by the co-receptor which is present over here, which is IHOG, which is over here in this diagram. So IHOG makes the binding of hedgehog and patched stronger. Now, as this is bound to the patched receptor, the patched receptor can no longer inhibit the smoothant over here, which is present inside the cell on a vesicle. You must remember that I said that the patched is inhibiting the smoothant, which is making it inactive, and the smoothant was not able to do all its function, that was all the downstream processing of the hedgehog signaling. Now since patched is occupied by hedgehog, it can no longer inhibit smoothant. And what happens is, once hedgehog binds to patched, this whole complex is internalized and moved into a compartment called the lysosome. And lysosome contains a lot of hydrolytic enzymes or hydrolysis. So what these hydrolysis do, they degrade the whole complex of hedgehog and patched, including the IHOG, and it is degraded and released into the cell. So the main thing is that the patched, when hedgehog is present, is not able to inhibit the smoothant. So what happens? This vesicle moves up to the membrane of the cell and fuses with the membrane. This fusion of the vesicle with the membrane moves the smoothant. This is my smoothant. Moves the smoothant onto the membrane of the cell, and after this is on the membrane, this smoothant can be activated by phosphorylating using CK1 and pKa. So when it is moving on to the membrane, CK1 and PKA is phosphorylating. Now, as this is getting phosphorylated, this is getting activated. Now, this phosphorylated smoothant can call upon this complex, this costal 2 fused and cubitus interruptus. So it is doing that. It is calling on to this complex. It recruits this complex. And this binding of the complex of costal 2 fused and cubitus interruptus, what it does, it inhibits the phosphorylation of cubitus interruptus. It inhibits the phosphorylation of cubitus interruptus, and cubitus interruptus is made free from this complex. So binding on to the smoothant changes the conformation of the complex, which releases this cubitus interruptus, and this binding also inhibits the phosphorylation that was being done by the CK1, GSK3 and PKA. Now, as this is freed from the complex, this can no longer go into the proteasomal lysis. So now this can do its function. So that's what it does. It moves into the nucleus. And it acts as an activator for the genes that were supposed to be transcribed when hedgehog signal is present. So it does exactly that. It moves into the nucleus and it binds to those set of genes, or to those the promoters of those set of genes, and activates those genes. And those genes are transcribed, and that is formed into functional proteins, which can do their functions in the development. So once more, the hedgehog protein binds to the patched receptor, and this binding is made stronger by the IHOG, and after this is bound to the patched receptor, this can no longer inhibit the smoothant that was present on a vesicle inside the cell. What happens is it moves into the cell into a compartment called a lysosome, and lysosome, since it contains a lot of hydrolases, it can degrade this whole complex. Whereas on the other hand, the smoothant moves into the membrane of the cell and is phosphorylated by the CK1 and PKA. Now, as it is phosphorylated, it is recruiting the whole complex of fused costal 2 and cubitus interruptus. Since it is recruiting, it is inhibiting the phosphorylation of cubitus interruptus, which is making the cubitus interruptus free from the complex. And since it is getting freed and not phosphorylated, it is not going into the proteasomal destruction. And now the cubitus interruptus is in its full form. It is not degraded. So the full cubitus interruptus moves into the nucleus, where it acts as an activator for those genes that were supposed to be transcribed by the hedgehog signaling, and it does exactly that. So it makes this protein, and these proteins can go into the developmental process. So that was all about the Wnt beta-catenin pathway and also the hedgehog signaling.